Hi everyone, welcome back to the GEP video series. My name is Cece and I am one of the virtual TA with the GEP program. And today we will continue on part 3 of the pathway annotation walkthrough. In part 1, we mapped the genomic neighborhood around RAP in the melanogaster. Now we will examine the genomic neighborhood of RAP in the Yakuba, and then compare the order and orientation of these genes to what we found in the melanogaster. Based on parsimony, the genes surrounding RAP in the Yakuba should be identical or very similar in sequence to the genes in the genomic neighborhood of RAP in the melanogaster. Additionally, the neighboring genes should also match in the orientation. For example, look at the direction of transcription of the neighboring genes, and this is called local synteny. In the melanogaster, RAP is on the positive strand and CG2926 is on the negative strand. These two genes should also be on a different strand in the Yakuba. This image is a guide to help users interact with and customize the genome browser interface to analyze genomic data effectively. I will be demonstrating these instructions in the next section. In part 3.1, we will examine evidence for a protein coding gene in the region surrounding the T+N alignment in the target species. First, navigate to the Genome Browser Gateway page, and then click on the target species D. Yakuba in the UCSC species tree and connected assembly hubs table. Under the D. Yakuba assembly field, confirm that the correct assembly is being selected. If you are unsure, the Pathway Project Genome Assembly page shows which assembly to use while annotating. In part 2, we determined the putative ortholog of RAP-PA is located at approximately these coordinates on the NC052530 scaffold of the D. Yakuba genome assembly. We'll enter this information under the position and search term field to examine this region. And before we continue, keep these 4 items in mind as you will be asked for it in the annotation form. Now click on the Go button. In the list of the buttons below the Genome Browser image, click on the default tracks, and then we'll zoom out 3x to get a better look. Now on the Genome Browser page, we should see several tracks, including the NCBI RAF-seq genes, the spline alignment of the melanogaster proteins, and also several gene prediction tracks, including GMOMA gene prediction with RNA-seq, the NSCAN PASA-EST gene predictions, and the Augustus gene predictions. And after that, we have the aggregated RNA-seq coverage. Before we continue, each evidence track in the Genome Browser has an associated description page that contains a discussion of the track, the methods used to create the track, and in some cases, filter and configuration options to fine-tune the information displayed in the track, for example, the RNA-seq tracks. If we want to obtain more RNA-seq data on the adult flies, we can change the RNA-seq coverage stages to full and refresh. Now to view the description page, click on the gray menu button to the left of the display tract or on the label for the tract in the display control sections. Now there are several ways to interact with these tracks. For example, zooming in a specific region, you can click or drag in the base position track to zoom in the highlighted region. The order of the tracks can also be changed by dragging the tracks up and down using the gray menu button on the left. The browser can also be moved between left or right by dragging the screen to navigate to different positions along the genome. And lastly, for shortcuts, we can type the question mark on the keyboard to view the available shortcuts for easier navigation. And this summarizes this image, and you can use it to refer back to it. Looking back at the NCBI RAF-seq genes track, We notice that the alignment for the coding regions of the D-Yakuba-Ref-seq transcript for XM ending in 5862 against the scaffold NC052530 of D-Yakuba line up with the spline alignment to the D-melanogaster protein of Rep gene for both isoform A and B. Furthermore, the coding portion of the 5 alignment blocks are incongruent with the placement of the 5 coding exons predicted by Gemoma, NSCAN, and Augustus gene prediction tracks. According to the ref-seq transcript for XM ending in 5862, the putative ortholog of rep-PA is located at the NC052530 scaffold with the 5' end starting at 19150,510 and the 3' end ending in 19152,080 in the Yakuba. And the coding region is within the putative ortholog of the ref-seq transcript. And the coding sequence is part of the DNA sequence of a gene that is translated into a protein. Before we move to the next section of part 3, we have to caution that we cannot always trust what we see in the genome browser is accurate, particularly for Drosophila species that are more distantly related to D. melanogaster. The gene prediction tracks, like their name implies, are predictions. Thus, your role as a researcher in the Pathway project is to help scientists studying these genes be confident in the specific model for the gene. Your brain is far superior to a computer algorithm in weighing conflicting evidence. Thus, your model will be more reliable than what the computer can produce alone. For example, in your own project, there might be a situation where a gene predictor doesn't show a gene in an area that has an alignment to the D. melanogaster protein, or vice versa, and therefore you will need to investigate that further.